All right. Hello, everybody. Thank you for joining us and welcome to the launch of Aivia 7.5. My name is Quinn Tran. I'm the scientific liaison here at DRVISION, and I'll just get us started real quick before I hand it off to our presenters for today. So speaking of them, the two people who will be presenting today is first Luciano Lucas here on my right, who's our executive VP.
Hi.
His expertise is in microscopy, image analysis, and software development. On my left here is Mike Jones, one of our algorithm engineers, and his expertise is in machine learning and object tracking. So for today's agenda, we'll start really briefly with an introduction to DRVISION, just for anybody out there who hasn't heard of us before. We'll take a look at the development of Avia's first year. It turned 1 on April 19th of this year. We'll do the launch afterwards for Aivia 7.5, where we'll cover our new features, including the pixel classifier, the mesh cutting tool, and the live import. For the pixel classifier, we'll be talking about machine learning versus non-machine learning algorithm. And just so you know, the pixel classifier is a machine learning feature. We'll also talk about the architecture, the features, and the classification that goes into this new application. And we'll also give you just a snippet and some introduction and some applications that work really well with this pixel classifier. We'll then follow up all of this information with an actual live demo of all of these new features that we have for you. And then we'll end with a short Q&A session. So be sure to ask any questions that you have along the way. You can do this via the YouTube chat room, also on our Twitter, and also on our LinkedIn and our contacts page on the website. So with that, I'll hand it off to these two guys and let them get started.
Thank you, Quinn. Thank you very much for the introduction. So Mike will join me again in a moment. You will be giving us most of this part, or a big part of the pixel classifier presentation will be done by Mike. He'll join me in a moment. He'll also stay at the end to answer any specific questions to do with the technology we have in the pixel classifier. So thank you very much for joining us for the launch of Aivia 7.5. It was about 2 months that we were working on this new release, and we're really excited to bring you these 3 major features, and we actually have quite a few more. We can read about them on our website and on the release notes. But for now, let me give you a brief introduction of DRVISION, because I know it's still not as known as other companies in this space of machine learning and image analysis. So let me give you a little bit of a background. The group of people that make up DRVISION actually started working together in the late 80s, and they had a company called Neopath, which made a smart microscope, this automated pap smear screening microscope. And you can see it here. It's still sold today by BD Bioscience as Focal Point, it's called. So you can still buy it for your lab if you need to have a lab like this, and it's still being sold and still being used worldwide. This same group of people then established a new company called S-Vision, which was software specific, had no hardware development anymore, and that company was established by James Lee, who is our current CEO and president, and he's been the president and CEO of S-Vision and then DRVISION since 1999. In 2008, received some additional investment, and the company rebranded to DRVISION Technologies, and that's what it still is called today, and it continues to be focused on software development. About a year ago, April 2017, we launched a new product called Aivia. I actually joined about 6 months prior to that and started working on this product. And my background is in microscopy, tumor angiogenesis. That was my PhD was in that field. And in the last 10 years, I've been focusing on image analysis and software development for image analysis and visualization. So DRVISION is really a very interesting and very active company. It not only creates products that a lot of customers use, many of them OEM customers, but also it is very active doing R&D, and we have a very active pipeline of projects in different domains. And along the way, because we are creating new technology and creating new knowledge, we are also patenting and publishing the work we create here. In terms of machine learning, we have currently 11 patents in that field, and the first work we did was in the late 80s in that microscope, the smart microscope that you saw in the previous slide. We have since developed dozens of other applications and deployed them successfully out there, not only in life sciences, but also material sciences, metrology, and other fields. Specific to the pixel classifier, we have 4 patents that describe the core technology used in the pixel classifier that we'll describe today. And we have seen a major increase in the applicability of this kind of technology because computers are getting fast enough to use it in real time. And that's what makes it very exciting, is that you can apply these algorithms or this learning environment in a very efficient way. So from 2010 onwards, we really see that we can apply this kind of approach to many types of applications in a very quick way. So now we decided to bring it into Aivia, which is our flagship product, and yeah, we'll talk about that during the rest of this presentation. So before we go into this, let me just introduce the rest of the team here. We have Chi, who is one of the managers in the team, and Tuan and Lu. These 3 gentlemen, together with James, have been working together for nearly 30 years, and that brings a lot of stability, and a lot of knowledge gets retained here and built on over the years. The team is, however, quite a bit bigger, and this is just the Aivia team. There are additional people that are not working directly on Aivia that are here part of DRVISION, and they contribute to other projects, including R&D projects. But this is the Aivia team. On the top row and most of the second row is the software engineering team. Then we have the algorithm engineering team that Mike is part of. And then the customer-facing team that starts here with Kevin. We have Quinn, which you've seen a moment ago, Hoyen, who is the product manager, and myself. By the way, we are hiring right now, so if you want to join the Aivia team, we have a position as a bioimage analyst / business development manager for the East Coast of the USA. So if you are interested in joining the Aivia team, please do contact me. Either on LinkedIn, there's a job posting there, or directly by email. My email is luciano.l@drvtechnologies.com. So we have just reached a landmark of the first year of Aivia. So we've had a very active year in terms of product development, and we took a moment to have a break. We went to one of the mountains. We are based in Seattle on the west coast of the US, and around here there are really a lot of very beautiful mountains where people go hiking quite frequently. And we took a morning. We went very early morning and had a beautiful hike. There are quite a few waterfalls. These are just some of the sights that we saw that day. And most of the people on the previous slide actually managed to join this beautiful hike. So that was really a very, very pleasant activity that we had. In terms of product development, this is what we've achieved in this last year. We launched April 2017, and the key features, and I won't describe this in any amount of detail, just the key features, because we have other webinars where you can look at the details, and the website also. But the key features were VR for image exploration, not just volume rendered, but surface rendered, so you can look at results. We also introduced a fast neuron tracing algorithm and classification using machine learning of objects. We then expanded on both of those, that's why it's 5.1. We added more functionality to the neuron tracer and more options to the object classifier so that it can classify more types of objects into the end 3D. And also, we added a new recipe to detect 3D objects that aren't neurons, so blob-like structures. Then we took quite a bit of time to develop Aivia 6, several months, I think 4 months or 4.5 months. And that took that time because we were working on two major features. One was the first and still the only commercially available deep learning deployment for image segmentation of electron microscopy images. And we focused on detecting neurons and dendrites. And that's the picture you see on the bottom. And we also introduced a brand new volume rendering engine, state-of-the-art and capable of rendering very fast, very quickly, extremely large data sets. And now we're actually using the same volume rendering for the live import that you'll see in a moment later on this presentation. With Aivia 7, which we previewed at ASCB, where we had a poster, that scientific poster that showcased all of the key technology that we developed in order to launch this product, we introduced object tracking and lineage tracking, as well as a brand new mesh or surface rendering engine. So we had already created a volume rendering engine, which was capable of doing rendering very large images in volume, so voxels. And now we have a very efficient way to render many detected objects, many thousands, tens of thousands of objects, in fact, per time point. And now we're going back to our roots with Aivia 7.5 to use a very efficient way to enhance images using a pixel classifier. And that's most of the presentation today is about the pixel classifier. And this pixel classifier is a fast way to teach a classifier. It's unlike deep learning, where you may need hours to train an algorithm. In this case, it takes seconds. And so it's a big distinction between those approaches. And this is a content-aware feature enhancement. That's why we're calling it that. It's aware of the features that you have in there in the image, and we can enhance those features depending on the training that is provided. We have a few other features, part of 7.5. You can see them here, and I recommend that you read them in detail on the release notes and on our website. And today we'll primarily talk about 3 of them. And they are the mesh cutting tool. It has 2 modes. I'll talk about that at the end. We'll talk about the pixel classifier, which I've mentioned already. And finally, the live import, which is just public. It's a public beta. We're not yet releasing this as a fully fledged product, but we're very happy that we have it, and we want you to have it. We know a lot of you would want to have this next to your acquisition microscope or acquisition computer. So we are making this available as early as possible. So let's get right into the key part of this presentation today. We're talking about machine learning powered pixel classifier. And that's the tool that allows us to go from an image like the one you see on the left, which in this case has just one channel and multiple structures. And we can enhance those structures by simple paint strokes to get an image like what you see on the right-hand side, which can easily be segmented. Aivia, so before I get into it, Aivia already has 2 other types of machine learning powered tools. One of them is the object classification, which I've mentioned and mentioned just a moment ago. And you can classify spines, cells, dendrites, dendrite segments, dendrite trees, and whole neurons, and just regular cells, so blob-like 3D structures. We also have the deep learning image segmentation tool, which we launched with Aivia 6, and now we're launching the pixel classifier, which helps enhance images. So it's kind of at the early stage of the pipeline for image analysis. If you want to learn more or recap on what we launched, I recommend these 3 webinars. The first one was a year ago, was an introduction to object classification. Then we have an in-depth webinar about the technology that is used to create object classifiers, and we provide quite a lot of examples. And finally, the Aivia 6 launch, which has a 20-minute or 25-minute section on deep learning uh, history, an overview, some of the key applications, and then it goes into the deep learning, specific deep learning architecture that we've used in Aivia for the segmentation, automatic segmentation of neurons in electron microscopy, micrographs, 3D. So why all the fuss about machine learning in the recent years? Well, there are good reasons for it. I should also say that machine learning doesn't solve all the problems. In fact, in some cases, it's counterproductive to use it. But we already have in Aivia let's say the regular image processing methodology that is based on histograms and thresholds. And we are adding a layer on top of that, which is infused with machine learning capability. And so it makes sense to explain why we're doing that. So the big allure was first described when the term was coined. Arthur Samuel in 1959, this is a US national that was doing research in this field, and he described it as a field of study that gives computers the ability to learn without being explicitly programmed. Now, this is very attractive, maybe not for programmers per se, because it could take their jobs eventually. Just kidding. But it is very attractive because you can provide lots of data, and the learning architecture that you've defined, of course defined by the algorithm engineer, will fetch the key information that is in there and fetch the key patterns and create a decision based on that, or a prediction based on that. That's of course very attractive. That's Arthur Samuel. The interesting thing is, even though we've made huge advances in many areas, and machine learning is being used in a wide range of fields from finance to medicine, it's not yet fulfilled this desire to get the machine to learn for itself. It's not fully, it hasn't come to fruition yet completely. And this is a recent article you can see here from 17th of April this year that says machines will soon be able to learn without being programmed. In fact, in some instances, machines are already capable of doing this, but not across the board. And generally, and certainly general AI doesn't yet exist. So an AI that can generate other AIs and that cannot distinguish from real humans. So we're still far away from this, but we certainly have enough technology to help with image processing and help a great deal. So if we compare machine learning versus non-machine learning image analysis, there's quite a few ways you can look at it. And here are just a few examples. I have one slide here and one later that look at slightly different perspectives on the same topic. So with non-machine learning algorithms for image processing or image analysis, the programmer or the coder needs to create the exact set of rules that will be applied. And these rules are hard set. They are not changeable by the data. The data doesn't change it. The user cannot change it. The rules are set in stone, well, in code in this case. Then the user can provide input within those rules. And that's what these sliders are for. You have a few parameters that you can control using these sliders or the numerical input that you have here. And what happens then is there is a detection that happens based on the parameters you provided that are within those rules. So the data you've provided or your expert knowledge cannot change the detection algorithm or the enhancement algorithm. And then you can run this script essentially that runs through all these functions that are preset, and it produces a result, which you may be happy with or not. And then you can again adjust. And that's why previews are so important. You can have previews and you can adjust things, see how it looks, adjust again, and in some cases, this is the best way to get to a result. In other cases, where the images are a little bit more complex, more challenging, or there is a lot of noise, and we'll talk about the examples, then it's very hard actually to do the segmentation using straight non-machine learning algorithms. And it helps a lot to have either a preliminary step where you enhance the image, or a whole pipeline where you do the enhancement and the detection and the filtering all using machine learning kind of environment. So with machine learning, what the software or our algorithm engineer will do is create a learning framework, which has enough capability to extract the key information from the images without having hard set rules. Then the user, with his or her expert knowledge in biology or in metrology or whatever the field, will teach the algorithm by providing painting areas or regions that are painted either a line or a big paintbrush or a region. And those are the teaching areas that are assigned to particular classes that the user is interested in. Let's say living cell versus dead cell. That's a simple example. And then the software will take that input and will learn from the features, the intrinsic features of that image, plus the teaching that the user provided, and it can then predict the likelihood of each pixel being part of class 1 or class 2, or more classes if there are more classes that have been taught. So it's a prediction or a prediction of the probability of a pixel being part of one of the classes that was taught. So it's very dynamic, and it's very able to detect an enormous range of biological elements. So I'll stop here, and I'll get Mike to join me to go into the detail of machine learning a pixel classifier. So let me get him here.
Hi, so I'm Mike. As introduced, I'm one of the algorithm engineers at DRVISION. So I'm going to be talking about how the pixel classifier works. So previously in Aivia, when using most of our machine learning, you would interact by selecting measurements that you could use to classify objects. So for example, by this, maybe to classify a neuron, we'd be using the length, diameter, volume, etc. to describe the neuron. And then these representations would represent real-world measurements of the objects. So with the pixel classifier, for each pixel, the length or the width doesn't make much sense because each pixel is going to be having exactly the same measurements. And you could just use the intensity, but that's very limited in terms of the amount of information you could get. So with the pixel classifier, we introduced the concept of using features in machine learning. So for example, an example of a feature could be a Gaussian blurring. So for each pixel, or for the image, we're going to blur it with a Gaussian filter. And then at that pixel location, that is the feature, the result of the Gaussian blurring. And so these features describe either the intrinsic image properties or the object characteristics, etc. And these features can be used to enhance different regions of the image differently because these features might respond differently in different places. So the pixel classifier is following a traditional machine learning workflow where we specify manually the features that should be used and the scales, and then a classification is made based upon a classifier taught to provide the class examples. So there's two main phases of the pixel classifier on the algorithm side. There's the training phase and then the applying phase. So during training, there are class examples that are drawn by you, the user, and then also training images that you're working with, and then the feature types and scales that you've selected. So the training images and the feature types are combined to calculate the features for the image. And then the class examples are used to specify which features belong to which class. And this result of this is used to train a random forest, and we save that random forest off. And then now when you're applying, the test images come in and the feature types that you previously used are calculated, and we apply that trained random forest to produce either a classification or a class confidence. So for feature extraction, different feature types and different scales can be selected from the UI to extract the different pixel properties. So for example, the feature for each pixel will be extracted over an area centered around the pixel, its location. So for example, if this is the individual pixel that we want to extract features for, we'll be looking at a region larger than that pixel, and how large this area should be is specified by what scales are selected. So a smaller scale is going to select information very local to this pixel, but a larger scale will capture a much larger region and more kind of low frequency information. So then it's also important to point out that some features will extract multiple measurements for each pixel. So for example, the Hessian feature will extract multiple measurements. So for the Hessian feature, we create the Hessian matrix, and then we also extract the eigenvalues from this feature. So for a two-dimensional case, there'll be two eigenvalues. Three-dimensional, there'll be three eigenvalues. So we want to also not just use one eigenvalue, use everything that we can, and allow the classifier later to pick what is the best feature. So then also if multiple channels are selected, we use unique measurements for each selected channel. So for example, if you have different fluorescent staining, we'll use all of the channels that you tell us that we can use, and allow the classifier to pick what is the best features from each channel. So in this example, if this is the pixel, we're going to be creating a feature vector from all of the features and scales. So for example, in this case, we'll grab the feature from this Gaussian blur, grab this feature from the Hessian, and this from the Sobel, and then concatenate them together to create a long vector. So about the different feature types that can be used in Aivia. There are many different features that can be used, but we recommend starting with the Gaussian blur, difference of Gaussian, structure tensor, and Hessian features. And the reason that we say this is because these are very powerful features that are also very optimized to work well together. And also these features extract a wide array of information. So if this is the original image, the Gaussian blur is going to be extracting the neighboring pixel intensity at different scales. And then the edge properties are going to be extracted from the difference of Gaussian and the structure tensor. And then line, blob, and plane parameters will be extracted from the Hessian. And so in this case, you can see the different bright dots are corresponding to different blobs in the image. And then finally, when classifying tube-like objects, we recommend testing other features like the membrane projection or aggregated Gabor filter features. So this is an example of the Gabor aggregated features. And they'll look very synthetic, but they're kind of describing the different edges of the image. So for example, this small edge right here is the same edge as here. So also most of these features are two-dimensional. However, we've also implemented some of the most important features in 3D. So when using 3D, the size of the 3D kernel will then be scaled by the Z resolution to ensure that it works well for your image. Then also, when a combination of 2D and 3D features are used, these 2D features are applied in 2.5D to allow you to combine all the features that you want to use. So for classification, after the features have been computed, the pixel classifier trains a random forest. So if you don't remember or are unfamiliar, a random forest is a collection of decision trees, each taught using a random selection of pixel locations and feature measurements. So if this is our feature vector from before, this feature vector is going to be sent to the different trees of the random forest. And each of these trees is going to be processing this feature vector slightly differently because it was taught different from all of its neighbors. So in these trees, we have the decision nodes, which make a decision on a particular feature measurement, and then the leaf nodes, which indicate which pixel which class the pixel belongs to. So in this case, the feature vector will process down and then in return that it belongs to the yellow class. This is the red class and the yellow class. And all of these results are aggregated together and either produces a majority voting, so in this case it would be the yellow class, or the class confidence, which would be 2/3 yellow or 1/3 red. So if most of the trees are indicating that the class belongs to the yellow class, then you have more confidence that that pixel belongs to the yellow class. But if it was a very even split, then it would be uncertain. So for majority voting results are going to be displayed during the preview, and then the class confidence results are going to be output when applied. And so the main reason for this is that it's easy to go from the class confidence back to the majority voting, but you can't go from the majority voting to the class confidence. And so when it's output, the output confidence will just be 255 times the class confidence. And so now Luciano is going to rejoin me. One moment.
Yeah, thank you, Mike. That was really nice. It's a good refresher also for those of you that didn't watch the webinar with Cyan almost a year ago now. Yeah, a couple of questions. So you mentioned here majority voting results are displayed during the preview, and then the class confidence results are output when applied. So these are two different things. And I know some people will wonder, why are we taking different methods for the preview versus the actual output?
So the main reason for that is for speed. So you can compute the majority voting slightly faster than the class confidence. You can imagine if when computing the different trees, most of the trees indicate, or more than half in the two-class case, indicate it belongs to one class. You don't have to keep checking the other trees. You know that one of the classes already won.
Yeah.
So you can stop early.
Okay. So that's the way basically we can get to a quick or faster preview.
Right.
Because the other one may take a few seconds more. Okay, another question I had is, I know some people will ask, and we didn't cover it yet, is the number of classes. So in an image, typically there's at least background and foreground that you want, but sometimes there are many classes. You may have 5 or 6 elements. Like say you could see the mitochondria, and you can see the nucleus, you can see the cell membrane, and then the background. That's already 4 classes. Can we teach 4 classes?
Yeah, of course. So there's a limit of the number of classes, but it's kind of artificial. It's just 255.
Okay.
But the real main consideration is when you have different classes is just to make sure that you have examples for those classes.
Yeah.
And make sure that those classes look differently. So if you have 2 classes that look very similar, make sure you have more examples so that the algorithm has a chance to define the distinctions between the 2.
Mm-hmm. Okay. All right, good. So we're collecting questions via YouTube, Twitter, and LinkedIn, and just our regular website contact form. So we're collecting those, and we'll answer them at the end of the presentation. So I'll go on to the rest, which will include demonstrations as well of the pixel classifier, the live import, and the mesh cutting tool. So yeah, thank you very much. So Mike will go, but he will join us again at the end of the presentation in about 15 minutes. All right. So from a user perspective, how does this actually all look like? it's good to have an understanding of what is happening in the background. But from a user perspective, what you do is really you don't need to worry about a lot of what Mike just explained. It's good to understand it because you'll need to talk about it in your publication perhaps, or you need to talk about it when discussing the results. But when using the software, it's really unnecessary to worry about all the nitty-gritty. So from a user perspective, when you're using non-machine learning algorithms, you do have to set multiple parameters during the setup, as you saw those sliders. Whereas with machine learning, it's totally different. It's brush strokes, so very straightforward. The critical step with regular algorithms, let's say, is applying a threshold. When you press the button, it's applying all sorts of thresholds that are hard set, and that's the critical step. Here, with the pixel classifier, it's about learning a pattern or learning features from the image that are already in the image, and that's the critical step. And then deciding what is the best possible algorithm for a particular application normally would fall on it would be the responsibility of the user to decide, okay, I need to use this particular plugin or this particular algorithm that has these steps. And you need to know what those steps are, and you need to understand in detail what they're for. With machine learning, because the tool itself is evolving as you teach it, you don't have to pick a particularly correct one, let's say, because it's going to evolve. And of course, you can decide which features you want, and that can have an impact. But generally speaking, if you use our standard set, it will produce a good result. And you can, of course, turn them all on. The penalty there is it takes a little bit longer to train. But the key thing is it evolves, whereas the non-machine learning one will not evolve and will not adjust. So no surprise, the prior knowledge you need to have in order to use non-machine learning algorithms is pretty high. It's intermediate to advanced, whereas with machine learning tools, it's really low. You can start, and you'll see in a moment, you can start with nearly no knowledge, just knowledge about your image, about the biology, about the imaging, and that's sufficient. So this tool is a fully featured tool. We have added, you can see here are some of the features. I didn't put all of them here, but these are some of the items that we have and some of the things you can do. I'll just mention a couple that I find really, really useful. For example, that you can adjust the kernel size is really very useful if you have large areas that you're trying to detect. Let's say in a histopathology section, and you may have bands of tissue that are of a particular type. You then use a large kernel, and that's much easier and runs very nicely and creates nice channels that separate those bands. Whereas if you're looking for really tiny little objects like nuclei or filaments, then you can use the small kernel, and that will work better. Also, we have a very handy ROI for previews in case your image is really large. I've played with some images in the last few weeks that were 10K by 10K pixels from whole slide scanners, and those can be even larger, but if you have an image of that size, it helps to set some regions for preview when you're teaching and applying. So that's just some of them. There's quite a few, and you'll see them in action in a moment as well. So what's the workflow? Mike already hinted at what the workflow is, but here is it in a more simplified way. You load your image, you train the classifier, and then you apply the classifier. And when you apply it, you get a number as many as the number of classes you've trained and decided you wanted an output for. You get new channels. So if you have a single channel image and you train 3 classes and you decide you want three output channels, you'll get 3 new channels, 1 for each class. So the training process itself is pretty straightforward. You draw some regions, you get a preview. You may want to adjust or not the regions that you painted, or the kernel size, or the features that you've decided to use. And then you get a new preview. You can teach that classifier, and then you can apply it. And when you apply it, you get those additional channels that are related to the classes you trained. So going into the conceptual applications now. So what is this good for? Which is, I'm sure, a question that a lot of you will be asking. So this is good for a huge range of applications. I didn't even talk about specific applications. That's why I call it conceptual applications, because these could be anything. So I'll just start with the first one. In this case, we have a single channel which has fortunately or unfortunately some bleed through. So it should have been labeling just for the cell membrane or the gap junctions actually, but it has some additional labeling or bleed through in around the nucleus and very little in the cytoplasm. So that's what you can see. So in this case, we can train a 3-class classifier that will enhance or create a channel that is cytoplasm only, another one just for the nuclei, and another one just for the gap junctions or the cell membrane. So that's one. Another one is converting very low signal-to-noise images to high signal-to-noise images. And this applies to MRI images, to CT scans, phase contrast microscopy, and in this case is a DIC image. And it's really superb for this kind of application. Another one is where you have multiple channels. In this case, I have 3, so it's an RGB image, and it creates it has a brown stain in this area, which is an immunohistochemistry stain for a cancer marker. And we can teach a classifier to learn the blend of RGB in order to then segment those regions automatically. And you can teach the classifier and then apply it on a whole range of images to do that. So here in this case, we take 3 channels and we convert it into a single enhanced channel. Then another application, and I use histology because it's easier to show, but it could be fluorescence. So in this case, we have a H&E stain, and it shows multiple regions there. You can see blood vessels or blood pools in here. There are some other structures here. There's tissue, and there are nuclei. So we have actually multiple channels. So there's 3 channels as input, and I have 4 channels, or 5 if you count the background, as output. And then I can go and do segmentation on those and determine, for example, the surface area of the red, the blue, the green, all of these regions that have a biological meaning. And one final example is to convert images that have very granular or specular appearance, like this. It's a nuclei, a nucleus here, and the DNA has been labeled, but not the nucleus is not totally filled with DNA. There are areas with less or less compacted, and so the stain doesn't show as clearly. And you may end up with dark areas within the nucleus. This can be a problem for segmentation because it will start to partition things in the wrong way. So with the pixel classifier, you can, let's say, homogenize that nucleus so that the whole nucleus is nice and even, and then you can much more easily segment the images using the regular algorithms. So here's some examples now in video format. Here's the teaching, and there will be a live demo in a moment, but I wanted to show you this. So here I'm teaching the second class for the cytoplasm, which I'm calling cell in this case, then the membrane, and another cool feature is you can change with a shortcut. It's shift control and then the wheel, mouse wheel, and you can change the size of the paintbrush. And you can see it, I'm painting there the membrane. And after that, I will turn on the preview, and that's very quick. You get a preview. If something isn't good yet, you can just select whichever class you want to provide additional training for, and paint a little bit more. And that's what I'll do in the rest of the video. So by the end, it looks like this. So I hit teach, then I applied this particular classifier. And when I apply the classifier, it creates, so right now there's only 1 channel. It will apply and create 3 more channels, 1 for each of the classes that I trained. And you can turn them on and off to check what the results look like. So this is, in a nutshell, how to apply it. Let me give you a few other examples. So that's the single channel to multiple channel. This is multiple channel to single channel. So in this case, we take the RGB as input, and I'm training on this brown area, and then on the background. And after I do that, there will be a preview, and again, I can teach. The buttons for teaching and applying are at the bottom, so teach and apply, and I'll get my channels. So the process, as you see, is really straightforward. Of course, in the background, all of what Mike is describing is happening, and there's quite a lot of work that needs to happen in the background, and we try to make it as fast as possible, as near real time as possible. But of course, the actual speed will depend on the computer you're using. So this is a DIC image, a time lapse. You can see, I'll just show you the areas I trained. So I taught on this region, I taught on these extensions and background, and then I go and do a little bit additional teaching of background so that it adjusts a little more. And once I'm happy with the preview, and I should say the preview is often not as good as the real output. Yeah, we will work on making the preview more realistic, but it's a trade-off, as you know. We want to make it fast. The preview needs to be fast, so it's not as accurate as the actual output. So you can expect a little bit better results from the actual output than the preview, which is a nice thing. So there it is. Very high contrast versus the original image that was very low contrast. Another image is this one that I mentioned the nuclei. So here we teach around or inside of the nuclei. You can teach a few of the small ones, a few of the big ones, so that we have enough examples. We're also going to teach the background in this case. And it helps. You see here, there's some teaching right in between. That helps quite a bit to get a nice separation between the two. Can teach and then apply. And in this case, it's applying to a 3D data set. So it takes a little bit longer because it's a stack, of course. It's 67 images. And then you can go into 3D space, 3D view, and look at both the original and the processed or enhanced channel. Show you a couple more. This is a heart imaged by MRI, and we can very easily teach a couple of regions. So here's the heart. Over here we have background also was taught over here and over there, and now we have an enhanced image, and we go into the 3D space. Actually, I'll use this one for the demo, so I'll show it to you in a moment in a little bit more detail. And this one uses a CT scan with the lung cancer right here. And let me get to it so you can see. So we're teaching where the tumor is. It's right there. Also, we teach some background around here, in this area, and in this area. And you can see this image it's very challenging because the tumor is of the same intensity as the rest of the tissue, or the same appearance as the rest of the tissue. And if you go into 3D space, you can't even see it. You can only see it in the enhanced channel that we created right there. We have a few tools in Aivia that are really useful for creating on-the-spot objects. So this is one of them, and that allows us to very quickly segment the tumor in 3D space. And we can do a more complex segmentation for every other structure in there and then show the tumor in context, where you see the lungs, the rib cage with all the ribs, and the tumor embedded in the lung tissue. Okay, so some examples. I will do the demo in a moment, and I welcome you to stay for a few more minutes because we'll do a demo with some of these images. So right now, Aivia is certainly infused with machine learning technology. We already had object classification, and and deep learning for image segmentation, but this is the full workflow. So you can now enhance your image when you get it from the microscope. You can enhance it using the pixel classifier that we just described. You can then segment and measure either using the regular algorithms that we have. I think now there's 15 different recipes for different applications from 2D to 3D plus time. Or you can use the deep learning recipe, which is specific for electron microscopy images, which are very challenging to segment automatically. Most people do it manually by drawing contours in Z, and this is extremely time consuming. So we have an automated way to do this. And then you can classify the objects that are detected using the object classifier, which we launched a year ago. And we also have something called novelty detection. And this helps you identify phenotypes that you haven't taught on. So maybe you taught 3 classes, 3 phenotypes on objects, and Aivia will compute also the likelihood of a particular object or all the objects belonging to a 4th class that isn't one of the 3 that you taught. And that's also part of the standard Aivia platform. Okay, so moving on to the mesh cutting tool. This is a relatively simple tool, but extremely useful. It's used, as the name indicates, to cut meshes into 2. And we have 2 modes. One is a way to cut that applies or creates a plane that is of the size of the image. So this is what we call infinite cutting plane. That's right here. And the other one is under the line cutting plane. So you draw a line here, and you select, of course, the object you want to cut, and it creates a line or a plane only around or near that object, the object that you selected on, not the other. So this is great in case you have many other objects around that you don't want to cut. And this one is in case you want to cut everything. There is a very handy undo redo, which I'll demonstrate, and it automatically will update all your measurements, including track measurements, which is really one of the key ways to use this is for editing tracks that have been incorrectly assigned to a particular object. You can go and cut the object and reassign tracks. And so this is also very important for lineage editing, and that's what we have. So this is what it looks like. So in this case, I have 2 neurons, and they're at one point connecting. This is a data set from Dan Bloodgood from the Cash Lab in North Carolina. It's a whole mouse brain, actually, but we have a portion of the mouse brain here. So this is how to apply a line. So you saw I just drew a line. I'll replay that so you can see it again. So here is, I can draw a line in any direction. So I draw a simple line there. I selected my object already, so it creates a box around the object that I'm going to cut. And then I can just click the button that says split selected, and it will split the 2, in this case, 2 neurons that are next to each other, and some of their dendrites are touching each other, so we want to separate them. Another example right here. So I apply a line. You can see there's a plane only in that region, and I apply, and it is now cut. You can also use the other mode, which is the infinite cut. And I think that's what I'll do next, is apply the infinite cut. So I'm going to draw a line or select an object, I think that one, and then draw a line across the whole sequence. So I'm separating the surface part, so the top of the outer part of the brain would have been here, and this is the inner part. So I'm going to separate the 2 with a simple line that cuts across all of the objects that are selected. And you can select multiples. This, of course, takes a little bit longer because it's cutting quite a few others, not just the single small one. And after that, they'll all be colored differently. So the ones on the bottom are now independent objects, so they have independent colors. And you can see that quite clearly there. I'm selecting a few of them. Our standard selection color is white. You can see that. And what I just did there was a undo. So if I go back a couple steps, you can see here I'm press undo, and they all become connected again. Okay, and that is for the interactive mesh cutting. That's the this very handy feature. And now we're coming to the end. So the live import is one of the exciting new features we have, which is not, in our mind, is not yet a fully fledged feature, but it's a really very interesting one. It is in public beta, which means you can download it and get it for free. We welcome your input, and it will probably stay like that for some time. We're not sure yet exactly how many weeks or months it will be. It depends also on how quickly we progress with it. And the key or the core of this feature is essentially letting you see the images that you acquire faster. It was initially intended for light sheet microscopy because the images tend to be very large and they sometimes even have to have some pre-processing before you can visualize them. So that's what we focused on initially, but actually it works for many other types of microscopes. So you set up your microscope acquisition software, whichever one it is, whatever software you're using. You hit the button that will start to acquire, and just before that you should hit the button in Aivia that says, please look for files in this particular folder where the files are going to be saved from your microscope. And as the images are being acquired, Aivia will be watching out for those files created on disk and will very quickly load them on and convert them to an Aivia TIFF file, which then it can render very, very quickly. That's the whole idea. So you can have your microscope creating images and Aivia in parallel rendering them in real time. So what is our motivation? First of all, we want to provide real-time quality control. So you don't do loads of acquisition and then processing. And then at the end, after spending all that time, you realize, okay, the images weren't good, or there was something wrong with my sample. It moved away from the field, or something wasn't right. You can also, if everything is right, you can actually start to interpret the results straight away because you can see it in context. You can see the 3D image in context and you can start to interpret those results. And finally, because we've added some functionality to fuse two views, it will simplify and speed up image generation. So we can basically, for microscopes like the dual SPIM or dual light sheet microscopes, where you have two orthogonal views, we can fuse them on the fly. So online fusing of two orthogonal views and rendering nearly in real time. So what features do we have? I already mentioned a few of them. First of all, view 3D data on the fly as it's acquired. You can fuse two orthogonal views pretty much in real time, so in a few seconds. And this viewer, the volume rendering that we're using, is ready for very large data. If you want to read more about it, you can look at our poster, which is on our website. The poster that we presented at ASCB explains the key technology that we put into the volume rendering, but it's all set up and performs really well for large data sets. You can also read about it or watch the webinar from Aivia 6, the one with the deep learning. The first part is about volume rendering, large image volume rendering. So that's the two parts. And it creates an Aivia TIFF, which you can then process in Aivia very, very efficiently. So what do we currently support? Currently, we support multi-file import, live import. So if your microscope creates series of TIFFs, so let's say a TIFF file per time point, that's fine, we can support that. Or if it creates a TIFF file per Z position, we support that too. If you use RAW or DAT, that is also supported. And we can support some other files formats, quite a few others, but these are the ones we are currently supporting. If you have a single file, however, this is a little bit more tricky, and we've spent some time to make this work for the SLD file format, which is the one from 3i. They make several light sheet microscopes, like the Lattice light sheet and the Marianas light sheet microscope. And with those, we support live update of the file. In that case, there's only a single file, in this case, a SLD file. And as that file grows, Aivia can read the updates and can continue to render in 3D and 4D those updates. And the throughput is around 6 gigabytes per minute. So if your acquisition is set at, let's say, 1 minute between each 3D time point, then we should be able to handle single time points that are up to 6 gigabytes. If your data set is only 2 gigabytes, then we'll do it faster in 30 seconds or so. So that's how it will work. But it is very close to real time, and I'll show you a demo right after this. So in summary, these are the 3 features that we covered today: the mesh cutting tool, the pixel classifier, and the live import. And I think now I will progress to the live demo, which is always a very nice part. It's my favorite part, actually. Where is it? It's here. Yep, here it is. All right, so this is a live demo of Aivia 7.5. You can see Aivia 7.5. This is from yesterday, and that will very likely be the build that you will also be using. So if you haven't seen Aivia yet, it has a very large window where it's a 2D display, or 3D if you have a 3D dataset. Like this one is a 3D dataset. So this is a real-time rendering of large-ish. This case is not an enormous data set, but it's pretty large. This is another one. Okay, so it has this 3D window. It has the display settings for the volume rendered part and the surface rendered part right here. And then at the bottom is where you have all the recipes that are used for image segmentation. We also have the pixel classifier, and that's where we'll spend a little bit of time right now. So I'll go and start to give you a little demo of the pixel classifier. So this is one of the datasets we looked at during the presentation. This case is the single channel to multiple channels. So I'll create one more channel here. So this is the want. So I want to call this, I could call this cell or cytoplasm. And this will be nuclei. And I'll have a third channel, which will be membrane. Okay, I can adjust the colors, so I'll change these colors slightly, just to be a little bit easier to visualize. Okay, and I start my teaching. So first of all, the cytoplasm, then I'll do the nuclei, and then the membrane. And I'll just hit preview, and that's my preview. I will teach it a little bit more so you see how it behaves. So I'll say that this is actually cytoplasm, and it will try to adjust. That's already pretty good. I don't really need additional teaching, I don't think. You can see that's what it looks like. So I can then teach this classifier. I select which channels I want to output, in this case all of them, and I then apply. That will create now new channels up here. So we have the nuclei right here, membrane, and cytoplasm. And with this image like this, it will be extremely easy to go and do image segmentation on those. With any regular threshold-based algorithm, it will be straightforward. And that's what we would do too. So let's see, the other example is this multiple channels to single channel. So we have RGB. I'm going to train this classifier just on two classes. One is the, let's say, cancer region or area that has the brown labeling, which is a marker for cancer. Okay, see this? As I mentioned, it's possible to change the kernel size, so I'm going to change it to medium, because the structures I'm actually looking for are relatively large, and I can teach a little bit more in areas where I see is not doing is not producing yet ideal results. I can go and teach it a little bit more. That's pretty good already. You can see you can further enhance it if you continue to teach. You can get it a little bit better. And once that is done, you can also select additional features, which I haven't in this case. Once that is done, you can do the same thing you did on the other example. You can teach it and apply it. One other thing you can do is save it and load back a classifier that you created previously. So I'll show you how that looks. So I'm going to get a classifier here. It's this one. So this is a classifier I did earlier, and I'm going to apply this classifier to this image. And that is the output. So you can see essentially everything that is not white currently is Cancer. Which is this region. It is possible also to control in the preview the transparency, and I'll show you that in the next example. Let me show you this example of the heart. So in this case, there is a heart here. You can see it's kind of present, but there are other structures around, so it's very it's not straightforward to get a good segmentation. So we'll go and teach this from scratch. Here it is. Say this region and this region, also here, and then background would be everything else over here and here. Can already get the preview. Okay, so I'll teach that, apply it, and this is relatively large Z stack, so it's 151 slices. So now we have the raw data, the enhanced channel, and we can go on and either render this differently so we can get a better sense of what it looks like. We can also use the object editor, which I mentioned already earlier today, to do a quick segmentation of this object. So I just clicked on the tool, I can segment it, and now I have a model of the heart. But hey, there's maybe this whole part here is not heart. This is probably a different organ over here. So we might want to cut that, and so we can do that using the new tool I just introduced today. So you can go and draw a line. Actually, I have to select it first. There we go. I'm drawing a line here, and I can then split, and that should split these two objects. And I can delete that one if I'm not interested in having it. Then I have my 3D heart segmented. Can go and explore it. By the way, we could just hit this button over here, and I'll put my headset on and go and explore this data set in VR, both the volume and the surface rendered image. Okay, so that's a very quick demo also of the cutting tool, the pixel classifier. Let me see if I have any other examples that might be interesting of the pixel classifier handy over here. That are different from the ones I have. Okay, I'll show you one more example, which is this data set, which is a phase, or actually DIC. I'll start a new area here. So teach this one. Make it a little bit smaller. Okay. And get the preview on. Oh, I didn't teach background yet. There you go, some background. Okay. And yeah, that's what the original looked like. So if you play through, it kind of gets longer, it's growing. And if we have the preview on, that's what it looks like. Then you can teach, apply it, and you have, in fact, I had already run this, so now I'll have even more channels here. But you will get the new channel, it's this last one that shows that highly enhanced image, very high contrast, which you can then use for segmentation. For very large data sets, it helps to have, and I'll show you one here for larger data sets. It does help to have the ROI on. So this is, so you see this one is 4,000 by 3,000 almost. And in this case, I probably would come into a region of interest like this. I start a new classifier, have the preview on, I set my region, you set it over here. Now you'll see there's a little blue box around it. And then I start to teach. And I'm going to teach it actually to detect nuclei. So here's some nuclei. I'll put some background also in there. And I now should have already the nuclei detected. So you can see that worked really well. I then teach it, and I can apply it. Just change this to blue. So the preview is matching nicely. Color. And yeah, so that's being applied now to the whole image. And I could, of course, save that. So that's this last channel that I created. This one here. So that's the new channel, that's the background. That's the channel I just generated from this image. And you can see the nuclei are all detected. Okay, all right, so that's the pixel classifier kind of short crash course. You can select multiple training input channels, as I mentioned. If you're applying, you can apply it to multiple output channels here. And this is where you would select the features that you're interested in, and the kernel size is over here. So pretty straightforward to use. I think I'll finish by showing you a brief demo of the live import. So how does that work? It works by starting it here. So File, Start Live Import. You would go, of course, you need to have, I don't have a microscope here, I should say. I don't have a microscope open, so I have some files on a different folder. And what I need to do, I'll illustrate that. So this folder right here is this folder here. So it's empty currently, it doesn't have anything. So I'm pointing right now, I'm saying, hey, Aivia, look at this folder, keep looking at this folder. And what I'm going to do is add some files there. So you see the folder is here, it's empty, Aivia is on standby. And now I would go onto my microscope and say, okay, start acquiring. And I drop a file in there. Another file, and I should say each one of these is 2.2 gigabytes. Let's have a look. So these are all now acquired. So this is a really fast microscope. I managed to acquire all of these images within 10 seconds, which is possible. So this is 2.5 gigabytes. And so Aivia is processing those. I'm going to move this over here, and you can see what's happening. It gives you a little bit of a report on what it's doing. It already actually created the first and the second channel are already there. You can see them. You can go to the first channel, time point. This is second time point, and now this is the third time point. So we already processed, in that little bit of time that I was talking, we processed and converted and displayed 7.5 gigabytes worth of data in those whatever few seconds. It does vary a little bit, so sometimes it goes a little bit faster, sometimes it goes a little bit slower, but that's roughly the speed you can expect is around 6 gigabytes per minute. Sometimes it goes a little bit faster depending on the hardware you have. And you can always see what it's doing right here. So it gives you a report on what it's currently doing. So right now is doing, it's completed time point 2 and it's reading time point 3. Ideally, for this to work really nicely, you have a couple of SSD drives. One that will be the one where you save your data as you're acquiring, and another one which is the folder or the disk that Aivia uses as its cache folder. So that's basically it. It will go through these, so it has now moved on to time point 4, and it has completed 2, so now it will show number 3, so it's there, and now it has number 4. So it's progressing, and you can of course look at it, interact with it, change the rendering mode, investigate it in 2D and in 3D. So now I'm in 2D. Sorry, 3D over here. This is a Drosophila embryo. Of course, all of you that are in the field would know straight away, but this is what it is, Drosophila embryo. And you can also check it out in the 2D mode. Yeah, it's probably trying to refresh because already on time point 5 over here. So it's probably going to try and finish the rendering, the 3D rendering, before it lets me go into the 2D mode. So there's a few kinks for us to work out, but we're looking to make this a very, very quick tool. It is already very quick, but it can be even faster and more feature rich, and that's what we're going to be working on. So you can investigate the image in 2D, you can go into 3D, and all of the time the software Aivia is trying, and it is actively fetching new files and loading them on. So now it has finished that time lapse of 1, 2, 3, 4, 5, 6 time points, and you can play through it and have a look at it. So there's the 6 time points are there, and you can start to do image analysis on it if you want. If you have a dual view microscope where you can spin a light sheet microscope, there is an option, and I should show that at the beginning here. So if I start, just pause this. When you start the live import, there is an option here for fusion. So you can fuse along a particular axis, and we have 2 possible algorithms. One is linear, so it blends from 0 to the end of the image in a linear fashion, or exponential, where the center, the middle part of the image, is exponentially blended. We have a detailed description in our Aivia wiki page, and you're welcome to go there and check it out in more detail, or just ask us questions, and we'll be happy to answer them. So this is the live demo of the live import. In principle, this could work also across networks in case you have your computer in your lab and your microscope is somewhere else. As long as you can point your computer to the correct folder, it will do the job and will let you preview the images that you're acquiring on your microscope, which might be remote on Aivia. This, of course, works well if you have high-speed internet. So internally, within your institute, probably works really well. If you're across the world, it will depend on the internet connection, so it might not work as well. Okay, so with that, I want to go back to the slides here and just acknowledge a few people. I want to give you a big thanks to Henry Hay, who is doing his doctoral work at the Morgridge Institute, and he works in Jan Husken's lab. He has helped provide ideas and input and test images for the live import, and we continue to work with this group to further expand the functionality of this tool, which we are very excited about. Also, Dr. Sebastian Streichen from the UC Santa Barbara, he has provided us with and allowed us to use some of his beautiful images, and the ones you saw on the demo are actually from Sebastian. So thank you very much for making those available, and we will continue to use those for testing and demonstrations. They're very beautiful and very high quality. Um, Also, of course, the Avia users, thank you so much for preferring Avia and using Avia. It's really a pleasure when we get to talk with you and learn about your experiences, and through that we can then create better tools for your own work. And last but not least, my own team, the Avia team. This is a enormous team effort. I had Mike today join me, and he will join me in a moment. But really, there's a big team behind the product that you just saw. We continue to develop it, and we'll continue to do so in the coming months and years to make it the best we can make it. So thank you very much, everybody on the Avia team. And with that, I recommend you to go on our website, download or request to download Avia 7.5, try it out, and give us feedback. If you want the live import, you can get it as well. It is free. Just contact us via one of the channels and we'll get a license to you in no time. I will invite Mike back and Quinn. We might have some questions that came in during the presentation. So let me get them here.
All right, so we did have a couple of questions come in, so I'll pose these to both Luciano and Mike here. So the first is for the pixel classifier and training it in 3D. Can we do different planes? Let's say the XZ or the YZ instead of just the XY plane.
So currently, no, it's just going to be the XY planes. And so also the drawings will all be made on the XY planes. We haven't made a good way to draw in 3D yet.
Can you say the question again? So 3D?
Training in 3D images.
XZ.
Yeah.
Oh, okay. Nope, not currently. But you can certainly train in train for 3D data, right? I mean, we can go through the steps,
Right.
and if you use the 3D kernels or the 3D features, then it will take information from Z. You can also train in time, so you can go to different time points and train in different time points. We didn't really show that, but that's all possible, not only in Z, but in different time points to create the same classifier.
If you were really interested in doing that, I mean, it would be possible just to flip your image around, and so that the XZ was now your XY, and then you could train on that.
Yeah, Gabby, I would be very interested to know what the application is of training in XZ. So we can maybe talk about that offline. But thank you for the question.
All right, so I'm just going to go down a couple more questions specifically for the pixel classifier. So you guys have kind of sort of touched on this, but can we teach on multiple images for the pixel classifier?
Multiple images, only with the trick that kind of Mike was hinting on. So if you append multiple images on a time lapse, then you can teach on multiple images. But that's one of the features that we currently don't have yet. But we'll add that in due course.
All right, so how about this with the paint strokes? Is there a specific number that you have to meet or a specific amount of features you have to teach Avia before the pixel classifier becomes really effective?
So there's no no requirement. Hopefully you can see when using the preview how effective or how many more places you need to add additional brushstrokes. Also, when you hit apply and you notice that some regions are incorrect, maybe that's a good place to add some more teaching examples too.
Oh, also you have undo.
Right.
which I didn't show, but if you teach something that clearly makes your preview worse, you can just undo it. And we also have an erase uh, ruler, or rubber, so you can erase parts of the teaching areas.
Okay. And so some of our users do other pre-processing for their images, one of which is deconvolution. Can you both speak to how the pixel classifier in this case is different from deconvolution?
Sure. So deconvolution is kind of different because you're going to be using a point spread function that you either have to measure beforehand or theoretically define. With the pixel classifier, you don't have to worry or know about that ahead of time. It will enhance your image kind of a different way by looking at different features instead of using the known knowledge of a microscope.
All right, so we do have some questions for the live import. So
Okay.
Luciano, you showed specifically a data set that was acquired on the light sheet microscope.
Right.
Can this work for confocal microscopes?
Yeah. In fact, it works for any microscope that creates 3D images. The criteria or the limitation is how are the images being generated. As long as the microscope creates files that fit into the supported formats that we mentioned, and we're of course going to expand the list that is supported. As long as it creates files in that format, it's fine. So the formats are either multi-file TIFFs, so one TIFF per time point, and then another TIFF for another time point, or multi-TIFF for Z, so lots of files for one time point, and then lots of files for another time point. And we have the nomenclature that is ideal for us, but Aivia is actually very flexible in fetching the nomenclature from the file name so that it knows what's the sequence. And then for single files, for a whole imaging session like the SLD and special tips, we have also created special support for those, especially for the SLD, where there is only a single file on disk, and that file is being dynamically changed. So Avia knows, we've programmed it to know how it changes, and we can update the renderer based on the new information that is being generated and put into those single files like the SLD. Yep.
Okay, all right, and then I think I have one last question here. Well, maybe two.
Okay.
For the mesh splitter that you showcase here.
Okay.
So, is there a redo and an undo button? Is there ability
There
to do
is,
that?
yes. Actually, I did show that. So there is. You can undo and redo everything, all the cuts that you make. Basically, it's just Control Z, Control Y, and you can undo and redo. And you can see that very clearly because the colors change. So it's very easy to spot.
And then when you are making all of these edits and these changes, do the measurements get automatically updated in Avia, or is this something that we have to do?
Absolutely. That's really critical because the key reason we designed or created this tool is for editing. Initially, it was for editing tracks. So when you have an object that gets to a point, and for some reason our algorithm detected one object, but really there were two cells in that spot, then the tracking it's wrong. The track will be wrong. So we have to go in and cut that big surface into two surfaces, and when you do that, it will update all the track information, and you can continue with the track editing after that.
Very cool. Okay, that's all of the questions that came in through all of our various channels. So again, thank you everybody for attending our live launch. Again, if you want to try it out or see more of the videos that was showcased today, please feel free to go on our website, contact us, see a lot of the videos and everything like that. This recording will also be posted very soon, so if you want to rewatch any of these segments, feel free to do so.
Yeah, the only address missing there is our YouTube, and actually we have Instagram as well, but this is our Twitter, LinkedIn, and our website. Remember, we have an open job position right now as a bioimage analyst, doing also business development. So if you want to join us, do drop me a line and check out this video on YouTube. Should be there in a couple of days well, the uncut version is there right now, but the cut version, which will be a little shorter and with less hesitation, will be easier to watch too. Will be there in a couple of days. So thank you very much for joining. Thank you very much, Mike, for helping with the presentation, and Quinn, thank you very much as well.
Bye, everybody.
Bye bye.